Okay, it's a great pleasure to introduce today's lecture. So, as you may know, we have this series for many years, since 2003, when the great American immunologist Charles Janeway passed away. We inaugurated this series of lectures, and many of prominent immunologists, especially some of them coming from our country, they were at certain point were given this special lecture. Charles Janeway made really very important discoveries in 90s, and I'm sure that our today's speaker will mention this. It's my great pleasure to introduce you lecturer, Professor Akiko Iwasaki from Yale University. So Akiko is Japanese who moved to Canada and studied high school and finished high school and then studied at university in Canada. So she is graduate from University of Toronto, so specializing obviously in the biological sciences and biophysics, biochemistry. And then she worked for a couple of years as a postdoc in the United States at NIH. And then she assumed a position at Yale University School of Medicine. And she's working there already, I think, for maybe 15 years, I believe. So Akiko is a specialist in mucosal immunity and also in the field of antiviral defense. So she's studying reactions of primarily innate immune system against viruses. And she worked for some viral systems which are very important for human health, such as Zika virus. And currently she is making her lab and herself are making very important contribution in the field of anti-SARS-CoV-2 immunity, and you will hear this in her lecture. So Akiko is full professor of Yale University School of Medicine. She's also member of the National Academy of Sciences, which is a really high honor. And she is also Howard Hughes Medical Institute investigator. This is also, for those of you who don't know, this is also very distinct and highly prestigious status in the United States. I have to mention that Akiko is also a spouse of our good friend, not of some of people in this audience, of Ruslan Medjitov, whom we know for many years. And Ruslan, by the way, was one of the first speakers in this Charles Janeway Memorial Lecture Series. So with this, I will give podium to Akiko. Akiko?
Yes, thank you very much, Sergey, for a very kind introduction. I'm really honored to be giving this lecture. So I'll share my screen. Okay. So as Sergey mentioned, I am a professor at the Yale University School of Medicine. I've been here actually 21 years now. Time really flies. And I had the fortune of meeting Charlie Janeway before he passed away. I overlapped just a couple of years. But even at that time, he really did make a huge impression on me. And the entire immunobiology department has been influenced and has been founded in the principles that Charlie Janeway laid out in immunology. And so we feel his presence, even though he's not here with us, we feel his presence almost every day, all the time. I just borrowed this picture from my husband, Ruslan Medjitov, who is here in the center. pictured with Charlie Janeway and this other guy you probably know also, Sasha Rudensky, when they were both postdocs with Charlie. And so he has made a very important impact, not only with his own science, but by training the next generation of scientists and the next generation and so on. And of course, I knew about Charlie long before I joined the department. Mainly from this paper that really kind of transformed the immunology field altogether, which is really the discovery of innate sensor that instructs adaptive immunity. And you probably know the backstory of how Ruslan read a theoretical paper published by Charlie, and he had applied to join his lab, and miraculously Charlie accepted him, and the rest is history. So this paper really had a significant impact on the immunology itself, because until then there was very little link between the innate immune response and adaptive immune response, and that this paper really provided that link between these two systems. And so, for instance, some of the follow-up sort of work that I contributed with Ruslan are reviews that we've written together on the topic of innate control of adaptive immune response, either through toll-like receptor, or later on, there were many other immune receptors that were discovered, so that it became sort of sensing altogether by the innate immune system that regulates adaptive immunity. I've also contributed some insights on how viruses are recognized by the innate immune system that it is then used to instruct the adaptive immunity, such as the discovery of the toll-like receptors in the endosome detecting nucleic acids from incoming viruses. And also have done multiple other work on innate control of adaptive immunity by other methods, not just through innate sensing of the pathogen-associated molecular patterns, but also through controlling remodeling of the lymph node size by feed arterial remodeling. And that's mediated by innate recognition as well. So it's not just about dendritic cells recognizing the viruses or bacteria through innate sensors, but much larger contribution of innate sensing controlling adaptive immunity at the tissue level. So this is sort of how my research really connects to what Charlie and Ruslan have done together. And as I mentioned, many of the investigators at Yale are still continuing to pursue questions that were originally posed by Charlie. So he's really an integral part of us. So today I want to give you some updates on what my lab has been working on, in particularly regarding the COVID-19 disease. So COVID-19 disease is interesting because it has a huge variety of disease outcome depending on the host. And for instance, people have asymptomatic to mild infections while others suffer from fatal disease. We also have people who recover completely from the acute infection while others that have very long-term consequences. My lab has contributed to the understanding, for instance, that when you compare moderate, severe, and fatal cases in the hospitalized patients, there are misfiring of the immune response that happen early during the course of the disease, especially in the severe cases that involve effectors such as Th2 and Th17. As well as a lot of neutrophil and eosinophil involvement. And that all correlates with severity of disease. We've also demonstrated that the timing of immune response is critical, and it's not the magnitude of the immune response that's important to control the disease. We have also demonstrated autoreactive antibodies that developed during COVID, which I'll get into, and provided some evidence for neurotropic infection of the neurons that occur in patients and mice. We've also looked at sex differences in immunity and demonstrated that female sex is associated with earlier activation of T cells, which has a very good prognosis for milder disease, as opposed to male patients having more propensity to generate inflammatory cytokines early during the response. And that correlates with the worsening of disease. We've also looked at pregnant women who were infected with COVID. And in one case report that demonstrated that the virus can actually infect the cells of the placenta. And in another report, we demonstrate that the respiratory infection of pregnant women results in significant impact within the placenta, even though there is no local virus infection. And then that can mediate some pathologies. So pregnancy is one of the high risk factor for developing worse COVID symptoms. So today what I'd like to do is to give you some examples of what we are doing and what we've done regarding, for instance, the neutralizing antibodies, as well as different variants of concern that impact immunity generated by vaccines. And then I will come to talk about a mouse model of COVID-19 that enabled us to determine the effector functions that are needed to control SARS-CoV-2 infection. And finally, I'll give you a little bit of flavor as to what we're currently working on with respect to long COVID. So the first study was led by two brilliant trainees in the lab, Carolina Lucas and John Klein, who looked at the role of neutralizing antibodies in COVID disease outcome. And all of the human and translational immunology work that I'm about to share with you today was enabled by the Impact Yale Biorepository, which was launched by Professor Albert Koh in the School of Public Health. And this enabled us to collect biospecimen from patients who are in the hospital in cohort 1, patients who are outpatients, as well as healthcare workers who were provided a very nice sort of negative control. But as some of them became exposed, we were able to provide the initial results from their NP swab and saliva as to the presence of virus in these healthcare workers in order to rapidly inform them and so that they can isolate themselves. This was prior to vaccines being available, so it was really the only means of control. And we were able to collect a whole variety of biospecimen over time, providing us with longitudinal insights into the immune responses in these people. So what Caroline and John did was to measure antibody response against the spike protein, or the receptor binding domain of the spike protein, in people who are not infected, the negative control in the black dots, versus those who are infected but without having to be hospitalized. So these are the non-hospitalized individuals. And then you have the patients who have moderate, severe, and lethal disease in different shades that are indicated here. And so what we notice immediately is that the level of antibody correlates with the disease severity, and that the lethal COVID cases, there was a huge variation in the amount of antibody that's generated against the RBD or the spike protein. So when we looked further into these patients, what we found is that the nasopharyngeal viral load in these patients who died of this COVID-19 were elevated and maintained high over the course of disease, whereas people who are discharged alive from the hospital were able to control the viral load over time. And when we looked at the viral load at maximum antibody titer within each patient, what we saw was that the diseased patients had much higher levels of viral load at their maximum antibody titer, whereas discharged patients had lesser amount. And also we found a few high neutralizers who had high levels of antiviral antibodies to begin with. So these people are able to control very quickly the virus. And so when you follow these patients over time for their antibody, let's say the IgG against the spike or the RBD domain of the spike, what we found was that there's a significant delay in the increase in these antibodies in the deceased patients compared to those who are discharged alive, who are in the green line here. And the high neutralizers, as I mentioned, they start off with pretty high levels of antibodies to begin with, and they maintain high levels throughout. And so also notice that the maximum level of antibodies is actually higher in the deceased, but the deceased take a much longer period to get there. So then what Carolina did was to divide the patients into early neutralizers who developed the neutralizing antibody titers prior to 14 days of disease onset, whereas those late neutralizers developed the neutralizing antibody after the 14th day, and follow these patients over time. What she noticed was that the patients who had earlier neutralizing antibody titer were able to eventually control disease score. So disease score of 0 is discharged and 6 is death. Whereas patients who had late neutralizing antibodies continued to develop progressively worse disease, and many of them had lethal diseases. When she looked further into the demographics of these patients, what she found is that the early neutralizers and late neutralizers had no significant differences in age or the BMI, and there were no significant differences in the distribution within male or female. However, there was a higher levels of mortality as well as higher level of viral load in the late neutralizers. So this study demonstrated that the COVID patients with lethal disease have delayed antibody onset compared to those who were discharged alive, and the maximum antibody values were not informative in distinguishing these patients. And somehow this ability to develop neutralizing antibody prior to 14 days was associated with a better prognosis than those that developed it after that time point. And this study has implications for therapies like the convalescent plasma and monoclonal antibodies, because what it suggests is that the late disease that happened after 14 days of symptom onset may no longer be mediated by the virus itself, but may be driven by the immunopathology. So these kinds of therapies should be given early, and that's what exactly the clinical trials are showing. So now that we've learned something about the immune response in infected patients, what about vaccine-induced immunity? So this study really asked about how the variants of SARS-CoV-2 affect vaccine-induced immune protection. And this was, again, led by Dr. Lucas, as well as a postdoctoral fellow in Nathan Grubel's lab, Dr. Chantal Vogels, and Dr. Incy Eldrum is a pediatric infectious disease physician scientist. The three led this study along with many co-authors. The study design for this was to recruit healthcare workers who are either previously infected in green here versus previously uninfected in purple in equal numbers. And these participants were then, the blood from these participants were collected prior to vaccination and 7 days, 28 days post first dose, and 7, 28 days, and 70 days post second dose of mRNA vaccine. The good news is that the mRNA vaccines, especially after the second dose, elicited very robust neutralizing antibodies in everyone. So you can see that the baseline, there are some people with some existing antibodies, and these are the ones that had a prior infection. And these people with prior infection, even though they had antibody, the first dose of vaccine really increased that level of antibody, and that was maintained throughout the course. And people who had never been exposed to SARS-CoV-2 but were vaccinated also reached very high levels of antibodies against the S1, and the RBD domain of the spike protein. When we looked at the neutralizing titers, we saw similar things. That previously infected people started off with some levels, not everyone, some levels of neutralizing antibodies. And this was dramatically increased after the first dose in those participants. And then that was maintained high levels. Whereas people who never had COVID were also able to generate equivalent amount of antibody after the second shot. Except that we found that there are 2 people who never developed neutralizing antibodies, even though they have detectable levels of anti-spike IgG measured by ELISA. And we also demonstrated that the T cells, CD4s on top and CD8s on the bottom, were also generated very robustly by the mRNA vaccines. This is looking at 28 days post-second dose, against the spike protein of the lineage A, its original lineage. And also we tested a P1 variant, the gamma variant of the spike, and demonstrated a similar amount of T cell activation against these spike proteins for CD4 and CD8. CD8, there was a little bit reduced level of activation against the P1 variant compared to the ancestral strain, but still definitely positive response. We then turned our attention to the different variants that had circulated throughout the globe. Particularly, we isolated and purified and isolated lineages of viruses that were circulating within the community that included the original strain, as well as the iota and alpha variants, the delta, epsilon, kappa, gamma, beta, and so on. And we performed a neutralization assay using the sera from the volunteers that were vaccinated. And so against the ancestral strain, as I demonstrated just previously, many of them, except for 2 people, generated robust neutralizing antibody titers. And this titer was maintained across the variants until we got down to the Delta variant. So Delta, there was a significant drop in the neutralizing ability of the vaccinated people. And this corresponded also to the emergence of some of the mutations that were seen, such as the L452R mutation. So this Delta variants, as well as Epsilon, had some reduced levels of neutralization. But then when you get down further into the Kappa, Gamma, Beta variants, then we start to see even further reduction in the capacity to neutralize these variants. And that also coincided with combinations such as the Delta 6970 combined with E484K, as well as the some of the furin cleavage site variants that were found in these cohorts. And on the right, what I'm showing you is the estimated effect of each mutation as a single mutation in their impact from escape from neutralizing antibodies. So as I mentioned, the L452R mutation here has one of the single most impact. However, the different combinations of mutations also impact the ability of neutralizing antibodies. When we looked At the inter-individual variation, however, we noticed that there's a huge variation in the ability of a person to generate neutralizing antibodies against all of these variants. And if a person is able to generate very high levels of neutralizing titer against the ancestral strain, this was, in general, maintained across the different variants. However, if a person starts off with a lower level, then you see the drop-off in the neutralization as these new variants emerge. And so this really demonstrates that there, even though the same vaccine is given, there is a huge inter-individual variation in the neutralizing antibody. And when you divide these participants into the previously uninfected on the top or the previously infected on the bottom, you start to see that there is a significant difference in the ability of the neutralizing antibodies to block these variants. That the prior infection does give advantage to having higher titer of antibodies, as well as more cross-reactive antibodies against different variants. So this second study illustrated that the mRNA vaccines do elicit very robust antibodies and T-cell responses to the spike protein, and that the previous infection gives people even a higher advantage to generating these types of responses, even across different variants of concern. And that there are distinct mutations in the variants that confer different degrees of escape from the neutralizing antibodies. And this really underscores the importance of having viral genome surveillance across the globe to be able to not only understand transmission of each variant, but potential for breakthrough. And even though I mentioned that the Delta variant, is not as capable of resistance from the neutralizing antibodies, the reason that the Delta variants are dominating right now around the globe has to do with its higher transmissibility, as well as potentially higher levels of virus being emitted by the infected person. And it also has the capacity to cause breakthrough infection that result in just as high a viral level as those who are unvaccinated, at least during the early phase of infection. So this, basically stresses the importance of vaccination, but also further measures such as wearing masks and getting a booster shot may be very important going forward. So, I focused a lot on the neutralizing antibodies, but what about other adaptive immune components? This is the question that was addressed by Dr. Ben Israelo, who is an infectious disease doctor who works in the lab. And what he wanted to do was to determine the relative importance of T and B cells in protection against SARS-CoV-2. So there's a lot of very impressive data out there that correlates the protection to a particular effector function of the immune response. This study, for example, here demonstrates the very nice correlation between the neutralizing antibody levels and the protective efficacy of a given vaccine, as well as potential exposure to a virus and prior infection here. And it really does seem to track with the ability to protect a person. And then also there are non-neutralizing antibody functions that are associated with protection that involve the FC function. And then T cell obviously are not only important in generating neutralizing antibodies, but also as effector functions for controlling the viral load and preventing severe disease. So it's great that we're getting a lot of insights, but we also have the advantage of using a mouse model to interrogate which of these are important and the relative importance of B and T cells. First, Ben asked this question in the context of a primary infection. There, what he found using the model where he can use AAV encoding humanase 2 to transduce mice of any genetic background with humanase 2 that enables these mice to become infected with SARS-CoV-2. And you can measure viral titer, disease, and so on. And what he did was he used either wild-type animals or the MUMT mice, which are devoid of B cells. And then he enabled infection of these animals using a humanase 2 transduction, and then measured the ability of each of these animals to control the viral load over time. So what you notice here on the bottom is the infectious viral titer in the lung. It's completely eliminated within 7 days of infection in wild-type animals. However, in the MUMT animals, you see that this is slightly delayed. Within 7 days post-infection, you still have some animals that have detectable virus, but within 14 days, completely gone. So this demonstrated that B cells are not necessary to control primary infection. However, when the animals lacked both B and T cells, such as in the case of rag knockout mice, these animals were no longer able to control the virus and they were chronically infected, indicating that you do need adaptive immunity to control primary infection. Further, Ben started to interrogate the importance of CD8s and CD4s in primary infection. And again, using the AAV model, he determined the importance of CD8s and CD4 by depleting them prior to challenge with SARS-CoV-2. On the top here, on panel C, demonstrates the infectious viral particle in the lung at 14 days post-infection in animals that are depleted of CD8s and CD4s. And you can see that the single depletion of CD8s or CD4 had no impact on clearance of the virus, but it's only when he depleted both CD4 and CD8s, he saw some of these animals still having virus at 14 days. And this is due in part to the fact that the CD4s were necessary to develop good antibody response, as well as in that context, the role of CD8 T cells in eliminating infected cells. And in the MUMT animal, he did the same exact experiment. And so the MUMT animal, as I showed you earlier, who are PBS treated, are able to control the viral load within 14 days. However, when they were depleted of CD8s or CD4s, he saw some of these animals unable to control the virus within 14 days, indicating that in the absence of B cells and antibodies, that those animals in part rely on CD4s and CD8s for control. And depletion of both of these subsets resulted in inability to control virus in the MUMT, sort of similar to what he found for the rag knockout mice. He then wanted to understand the role of B and T cells in protection against reinfection and vaccine-mediated protection. First, he took an adoptive transfer approach where he collected either immune sera or T cells from animal that recovered from SARS-CoV-2 infection. And then he transferred these into AAV humanase 2 expressing rag knockout mice. And then he infected these animals with SARS-CoV-2 and measured the virus 7 days later. What this graph shows you is that those animals that were transferred with T cells, immune T cells, some animals were able to control the virus altogether, while others still had maintained infectious virus in the lung, indicating that T cells are partially capable of clearing the virus. However, the serum transferred animals, none of them had any infectious particle at this time, meaning that the serum antibodies are providing a complete protection against infection. Ben then took an approach where he created a situation that some animals had high levels of antibodies against the spike protein, while others had intermediate or low levels. And he did this by using K18 mice. These are humanase 2 transgenic animals that can also be infected with SARS-CoV-2. He took different doses of mRNA vaccine, 1 microgram all the way down to 0.01 microgram, And then he measured anti-spike IgG in these animals. And there's a beautiful dose-dependent response depending on the animal receiving 1 microgram, 0.3, 0.1, 0.03. And by the time you get down to 0.01 microgram, there's no detectable antibodies. And then he challenged these animals with SARS-CoV-2. And what he found is that all the animals that were immunized with 1 or 0.3 microgram of mRNA were completely protected from disease or death, whereas those that had only 0.1 microgram, half of them succumbed to infection, and anything less than that, all these animals succumbed to infection. So this provided Ben with the ability to calculate correlative protection based on the antibodies. So there's this beautiful correlation between the amount of S1 specific antibody generated in the animal and the prevention of weight loss at 6 days post-infection. And based on that, he was able to calculate the pre-infection S1 specific IgA that's required to confer a protection from death. And similarly, he calculated the same thing for neutralizing antibody titers. Finally, Ben wanted to know what is the role of CD8 T cells in protection against SARS-CoV-2 infection in the context of reinfection or vaccine-mediated protection. So he created 2 cohorts of mice, one's that's been immunized twice with the mRNA vaccine, and the other that's been recovered from a prior sublethal dose of infection. And then these animals were challenged with either the original strain, the Washington strain, or the beta variant, which has the highest escape capacity. And then he depleted CD8 T cells or not from these animals. The vaccine induced very robust antibody response, and so did the prior infection, vaccine being slightly better than prior infection. And all of the animals that were either vaccinated or prior infected were completely protected from disease or death, regardless of whether they were challenged with the wild type, the original virus or the beta variant, or they were depleted of CD8s or not. And when he looked at the viral load in the lung, he saw again that the vaccinated or convalescent animals were completely protected from infection, regardless of whether they were depleted of CD8s or not. So this study demonstrated that T and B cells can both contribute to clearance of the primary infection, and that in the absence of one, the other can compensate. However, when the animals are lacking both B and T cells, these animals are no longer able to control the infection and they become chronically infected. Adoptive transfer studies demonstrated the partial effect of T cells and complete effect of the serum antibodies in protection. And in the convalescent animals and in vaccinated animals, depletion of CD8 had very little impact on protection against disease or death. And antibody levels are correlates of mRNA vaccine-induced protection. So this study, along with many studies of human volunteers that's been vaccinated with the mRNA, illustrates the power of this particular platform for conferring some level of mucosal immunity. So there are studies now demonstrating that you can isolate neutralizing antibodies from saliva and nasopharyngeal swabs in people who are vaccinated with mRNA and indicate IgG and IgA isotypes, indicating that there is some crossover of systemic circulating antibody to the mucosal surface. And these types of antibodies are likely to be able to confer neutralization and prevention of infection. However, even if that were to fail, there is the possibility of the existing T and B cell mediated functions that control further replication and spread. And even if that were to happen, the vaccine reduces, obviously the spread and the virus into deeper lung and also symptomatic infections, which results in sneezing and coughing, which would enhance the transmission. And even if the person were to expel the virus through sneezing or coughing, perhaps those viruses will be coated with antibodies to make them less infectious in the next person. So this really demonstrates that mRNA vaccine to some extent are able to do all of these things. However, compared to a mucosal vaccine, the degree of this kind of sort of effector function is still much, much less. And we're currently developing mucosal vaccines in order to prevent not only infection, but transmission. So finally, I want to talk a little bit about our current and ongoing work on long-term consequences of COVID. And this is sort of, there are so many people, millions, suffering from long COVID. And people often think of long COVID as being the result of someone who's been severely ill and hospitalized. And it's true that these types of patients who are discharged from the hospital suffer long-term consequences, and large percentage of those people appear to suffer. However, it's also important to point out that mild or even potentially asymptomatic COVID can lead to long-term symptoms with no longer detectable virus in their nose. And this percentage has been, each study has different percentages because long COVID isn't defined uniformly by all the studies. Somewhere in the estimate of 10 to 30% of even mild cases turning into long-term disability. And so I'm going to focus a little bit of the talk on the latter. We're doing both kinds of long COVID studies, but mainly kind of focusing on the non-hospitalized patients for now. So in both cases, actually, there are over 200 symptoms that are associated, that have been reported with long COVID, and that involve multiple organs, including systemic impact, neuropsychiatric, head, eye, ear, nose, and throat, and endocrine, cardiovascular, pulmonary, musculoskeletal, and GI tract. And this is just a handful of typical symptoms that are being reported from these patients. And the long symptoms do vary over time in severity and duration. So the very severe and severe cases of long COVID do decline over time. That's replaced with more moderate and milder cases. However, the fact is that there's a flatlining of some of these moderate to mild cases in about 6 or 7 months, and people are suffering over well over a year in some cases. And when you look at the average time of recovery, those people who start off with a slightly less number of average symptoms tend to recover within 90 days, whereas those patients starting off with 13 or more symptoms evolve more symptoms and are not recovering. So we have a couple of hypotheses about how long COVID might happen. One is that viral reservoir, so infectious viral particles or viral RNA and or protein being lodged somewhere in the tissue that's not accessible by the NP swab, and that's sort of causing chronic inflammation. The other possibility is autoimmunity. It's possible that autoreactive lymphocytes, B or T cells, can target different tissues that would result in the multiple symptoms that I just mentioned. And it's also possible there's tissue damage, dysbiosis of microbiome, as well as latent viral reactivation, which is often reported in the long haulers. These may all contribute together. None of this is mutually exclusive. Some people might have all of these, others may have a subset of these disease causes. There's evidence for both types of the major hypothesis. The viral reservoir can be seen in COVID patients. This is an intestinal biopsy from a person that had recovered from COVID. And you can see that there is nucleocapsid staining within the enterocytes of this particular patient. And so survey of 14 patients, 5 out of 14 had positive staining for viral antigens, and 3 out of 14 had PCR positive for viral RNA. Another study demonstrated similar finding in the variety of GI tract organs, as well as in lymph nodes in convalescent individuals ranging from 9 to 180 days after testing negative for SARS-CoV-2, demonstrating RNA and or protein can be found in various tissues. We've also collaborated with Professor Aaron Ring, who's in our department, to look for autoantibodies in acute COVID-19. And this work was made possible because of a new technology that the Ring Lab established called Rapid Extracellular Antigen Profiling, or REAP, where his group generated yeast display library expressing human exoproteome, which are extracellular and secreted proteins. And at the time we did this analysis, there were about 3,000 of these exoproteome. Now he has increased it to over 6,000. And he was able to basically make a very high throughput assay where we incubate the patient's serum IgG together with this yeast display library. We pull down the cells that are bound by IgG using magnetic beads, and then we decode the antigen that's expressed by those target cells. And by using this REAP, not only did we identify a variety of autoantibody against immune cytokines and immune cells, but we also demonstrated a large number of autoantibodies in COVID patients across many different organ types. So each column is a patient and each row is an autoantigen. And you see that there are sort of a variety of reactivity that you see in a given patient against all of these different kinds of antigens that are listed here. So this suggests that these types of antibody, if they were to persist, might contribute to a pathological outcome that's seen in long COVID. And there are many other reports now reporting autoantibodies in both acute and long COVID. And this last paper here demonstrated that all of the patients with long COVID had at least one autoantibody against G protein coupled receptors, which are endogenous antibodies against G protein coupled receptors have been linked to many diseases, including autoimmune diseases. So are there any treatments that impact long COVID symptom? There is currently no great diagnosis or cure or therapy against long COVID. However, there's some hint that the vaccines might have impact on long COVID symptoms. This is a patient group called Long COVID SOS that reported that about 50 to 60% of their patients felt improvement in their symptom after vaccination, whereas about a quarter reported no change and the rest actually had worsening of disease symptoms. When they broke down the different symptoms, there were some differences with respect to the adenoviral vector vaccine, Moderna, and Pfizer in the impact of reducing fever, myalgia, and so on and so forth. However, overall, there was improvement in a large fraction of long haulers. So how can vaccines impact long COVID? Well, when you go back to the couple of hypotheses, the viral reservoir and the autoreactive lymphocytes, it's possible that the vaccine elicits such robust T and antibody responses that could clear the viral reservoir, in which case you no longer have any pathology. It's also possible that the cytokines generated by both innate and adaptive immune response can impact the T and B cells that are secreting toxic cytokines or antibodies, and that may temporarily dampen the PASC syndrome within these patients. And we don't know which of these things are happening, but that is sort of the target of our current study. So our mission is to try and understand immunological and physiological determinants of disease pathogenesis of long COVID. And this is really the only way to inform rational diagnosis and therapy. And it was generously funded by HHMI to be able to do this work. So what we're currently doing is we're collaborating with a large number of patient groups as well as different long COVID centers to isolate various biospecimen and analyze them in real time using some flow cytometry analysis. And the rest is being stored and archived for further analysis, including the REAP and also T-cell receptor sequencing and metabolomic and hormone profiling. And all of these data will then be analyzed in integrative and multimodal data analysis to determine the clusters of different symptoms in patients, and finally to understand if there's any good diagnosis or therapeutic targets that we can identify out of these types of analyses. And we want to share the data back to the patients and involve them as equal partners in this investigation. So I'm going to end here by thanking all the authors that contributed to the work that I was able to discuss with you today. And also my entire lab members who contributed so significantly during the pandemic to really not only try to understand immune responses, but also to help the community by doing PCR screening of the healthcare workers. And the Impact Yale repository, without which none of the human work could have been done. And many centers and patient groups that we're collaborating with on the long COVID end. And this is a recent picture taken after we were all fully vaccinated, and really happy that there are really great vaccines around the world. So I'm going to stop sharing and take any questions. I cannot hear you.
You cannot hear me?
Oh, now I can hear you. Yes.
So the first question is, all your neutralization assays, are they done with live virus or this is reduced system?
It's live virus.
On the cell line, right?
Well, what we did is we isolated and purified these different variants and used them to do the neutralization assay. And the reason we did this is because there is more than just mutations in the spike protein that may confer some advantage for some of these viruses. And that's why we decided to do it with authentic virus.
Thank you for a wonderful talk. It was really a pleasure to hear you. Where you are? Yeah, here. All right. I have a question. Do you have a data where, say, you observe the autoantibody after mRNA vaccination? Do you see this data? First question.
Excellent question. We actually testing this right now, and so far we haven't seen any autoantibodies with vaccines.
I see, thank you. And the other question, do you see the effect of virus like for exhaustion of immune system? For example, this viruses overstimulate the T cells and B cells as well, and maybe this fatigue syndrome which you observe is because of just maybe failure of adaptive immune system.
Yeah, that's another great question. So in the acute COVID, we studied very much the different T and B cell phenotypes, and Indeed, one of the striking things that happened during acute COVID is T cell lymphopenia. There's a significant drop in the number of circulating T cells that cannot be accounted for by their sort of sequestering in different tissues. So number one, there is this T cell lymphopenia that impairs the proper adaptive immune response in most people. And that is likely to have occurred in the long COVID patients as well. We don't have an acute phase of immune response from the long COVID patients because many of them weren't hospitalized and many of them, didn't have access to research like that we were doing. However, we know from milder cases of COVID that there is T cell lymphopenia happening even in milder infection. So that leads me to believe that there was already a defective adaptive immune response to begin with. And antibody response, as I showed you, is also not very well induced in mild infections. So there's T cell lymphopenia, reduced antibody levels, potentially contributing to the ability of establishment of, persistent infection. That's something that we're looking into, but we don't know. Unfortunately, a lot of these things require pretty invasive probing, like the intestinal biopsies, but we are, collaborating with investigators that are doing such studies. And so the T cell exhaustion and potentially the lymphopenic state may contribute to the ultimate outcome of long COVID.
If I may ask questions which is not directly related to your talk, since you are in the States, we are reading literature here. So tell us about this new pill from Merck. Does it really inhibits virus? I mean, what is the latest? Please update us.
It appears that it does inhibit viral replication, and the clinical trial data are pretty impressive. So, but it's not a replacement for a vaccine, of course. We still don't know what the long-term consequences of taking that molnupiravir. And so obviously it's best to be vaccinated. But if you do get a breakthrough, there are a couple of things that one can try, such as this new pill from Merck.
And what about remdesivir and the other things which were mentioned before? They are really, very, very low effect or no effect? Or what is now consensus?
Yes, unfortunately, the remdesivir, if you give remdesivir late in disease, there is no effect. If you give it early enough, there may be some benefit. And so, as I emphasized, it's really about the timing of immune response and or antiviral, right? So during early phase of the infection, the disease is likely still triggered by the virus infection, and therefore giving antivirals early would have benefit. But if you wait too long, the things that do work are steroids that would dampen the immune pathology. So, all the trials have kind of supported this view that the timing of the delivery of these agents is absolutely critical, even the monoclonals.
Thank you.
Thank you.
Andrei?
It's a question about mucosal immunology. So it seems like mRNA vaccines after the boost they induce IgG response in mucosal, right? So why is it basically IgG? So is it a passive transfer of the antibodies just because it's too much, or it's active transfer of the IgG antibodies, which we were not aware of?
Yeah, that's a good question. There seems to be some, passive transfer, potentially some serum exudate. It's all hand waving. I don't know exactly what's going on, but you can certainly find the IgG and a little bit of IgA in the mucosal secretions. And the dimeric IgA obviously requires a particular polymeric IgA receptor for transport, and that's likely, happening to very, very small extent with the mRNA vaccine. And if you're previously infected, there's a lot more dimeric IgA. However, the vaccine-induced antibodies, I think because there's so much in circulation, some of it gets through to the mucosa. And also, it's interesting, we're also trying to identify the difference between the saliva and the nasopharynx. There seems to be distinct levels of antibodies, at least, in these two different mucosa. So yeah, the transport mechanism and what's really driving this IgA presence in these mucosa, it's a very important question.
And then another question with regard to the boosting or not boosting immunity, right? Because now the society in a way talks about the levels of antibodies, and you sometimes get the impression that they have to be the higher the better, right? But then at the same time, we know that the reservoir and the immune memory is in a way limited. Do you think that pushing a system too much in one direction would actually open the new pandemic, a new option for pandemic when people will take off the mask?
Well, thank you for wearing the mask, by the way. But I don't know. I mean, so there's a fear, right, that people are talking about that once enough people are vaccinated, a new variant might emerge that can escape, the existing immunity, and they will be selected for because they have the sort of better fitness to transmit among a highly vaccinated population. We haven't seen that yet, so I'm just crossing my fingers that won't happen. But you never know. This virus, I mean, before the Delta emergence, I was a lot more confident about being able to control this pandemic with the vaccine. But with the Delta, there's a lot more sort of breakthrough infections, and not just the mild ones. And so, in the US currently, people are becoming more eligible for the third dose of vaccine. And especially if you're in the, 65-year and older, or if you're immunocompromised, people are able to get this after 6 months of the second dose. So, and there's also already evidence that the third dose is preventing infection and transmission. And there's no evidence that that's increasing the variants that are escaping such immunity. So far, so good, but never say never. I don't know.
So, Akiko, I want to ask another question. This adeno-associated viral system with ACE2, so how easy it is? Because we're also working on with transgenic mice, and of course, advantage here is that you can infect any knockout, any transgenic mouse without do genetic work. I mean, how easy it is?
Yeah, it's extremely easy, but I have to tell you that there's difference in pathology. So the AAV model is great for studying the immune response, but they don't get very sick. The reason is that the mouse pathology is mostly coming from brain infection. And in the K18 mice, if you do intranasal infection, you'll get both respiratory and some CNS infection. And it's the latter that's killing the animals. Whereas the AAV model, we're just transducing only the respiratory cells, and therefore they don't die of infection. When we transduce the brain, though, with AAV and infect them, in the brain, they die. So I think, depending on what you want to ask and what the questions are, the K18s are, more sort of conducive to pathology and death, whereas the AAV model will let you study immune response forever because these mice don't succumb to death.
Thank you.
It's me again. Sorry again. In the beginning of outbreak, right? a lot of big journals publish papers about case reports that some particular cellular populations, very important to have a mild symptom or low symptom, for example, B cells and T cells. Do you know some, like, population which actually really protect against of the load of viruses, not in terms of antibody, but in terms of like increase of this population or predisposition to increase this? Do you know this, such populations? Because I'm not directly from your field. Yeah, thank you.
Okay. I'm not sure if I quite understood your question, but there's definitely studies that correlate, T cell and antibody responses with milder diseases. And so, with the antibody, as I showed you, that it's driven by the severity. However, if you had a pre-existing T cell that can cross-react to SARS-CoV-2 antigens, which is shown by Alex Sette and others, that may confer some resistance. Again, like, it's really hard to correlate what was there before because we don't have a pre-existing data set for that. But there is also sort of kinetics of T cell responses being very important for recovery. So I think there's evidence that, of course, if you had very good pre-existing immunity generated by vaccine, that's like the best protective mechanism. But maybe you're asking about something else.
Yeah, thank you.
Thank you.
Hello, thank you for your beautiful talk. I have a question about the K18 mice, this model. So you showed some beautiful data on the mice that actually recover from the infection. And of course, we know that these mice are highly susceptible to this infection. So what are these doses? Can these mice possibly be used to study long COVID or actually to establish exactly what are the reservoir of the infection in vivo? Yeah, thank you.
Yeah, great question. We'd love to, establish a long COVID mouse model. The K18 mice, I'm not sure they would be the best model for the long COVID because they, if you infect them with the sublethal dose, they do clear the pathogen. And as I mentioned, the RAG knockouts that are transduced with human ACE2, they become chronic carriers, but in their case, there's really no pathology. So we're sort of trying to figure out how best to create a scenario in which we can mimic long COVID better. And maybe it's a combination of some immunodeficiency as well as having immune response that can trigger some of the pathology. So we're trying different models right now.
More questions? Please.
Thank you so much for your lecture. It was really great talk. My question is about, do you know a lot of scientists use a lot of variants of pseudovirus system and a lot of protocols for determine the neutralization activity of antibodies. What do you think about the correlation of these two systems?
Yeah, we thought a lot about that because the pseudotype virus is much easier. We don't need a BSL-3 facility to do the neutralization. Some of us actually, Ben, for instance, uses pseudotype virus for neutralization. It's just a lot easier to do it. One disadvantage of pseudotype virus is that, as I mentioned, there are so many other mutations that occur in non-spike region of the genome that may or may not be replicated well. Obviously, pseudovirus is only using the spike region. So it really depends on what the questions are. And there is definitely some correlation with anti-RBD antibody levels and neutralization levels, regardless of whether you use pseudotype virus or not. And there's also difference in sensitivity of the neutralization assay. So even within my own lab, there are different approaches to doing authentic neutralization assays using different cell lines and different culture conditions. So it's kind of like an art form, you optimize it for the question that you want to address.
Hello, thanks again for your lecture. I have a question dedicated to the slide to the slide about vaccines and long COVID. You said there that you have now two hypotheses of the emergence of long COVID symptoms. The first one being my question is about that one about autoimmune cells and how vaccines could deal with that. So you wrote in your slide that vaccines may improve long COVID symptoms by diverting autoimmune lymphocytes through innate and adaptive cytokines. How could that be? Could you explain, please?
Yeah, sure. So I'm just thinking of a parallel between the multiple sclerosis, which is treated with recombinant interferon beta. And so the way interferon beta probably acts on many different cells, but one of the target is likely the sort of autoreactive Th17, Th1 cell types. And it's known that interferon beta has immunomodulatory function against these cell types, and that stops their secretion of IL-17 and interferon gamma and so on. And it also has an impact on the blood-brain barrier. So that's what I was thinking, that there may be a parallel thing going on with the vaccine, that it may induce some of these immunomodulatory cytokines that can either act on the tissue or the T cells that are impacting, or it's potentially B cells, impacting these tissues to dampen the inflammation caused by the autoreactive cells. But we currently don't have any evidence. These are just hypotheses.
Thank you for a very interesting talk. My question is, how high is the risk of the newborn child to be infected when mother is already infected? Is it 100% or is it not so?
I don't think there is any evidence for vertical transmission. So if the mother is infected, she may even be able to transfer some antibodies to the babies. But it's not that the baby is guaranteed to become infected. It depends also on the stage of infection, right? So if the mother had infection way before labor and recovered, that's a situation in which adaptive immunity might help the baby. Whereas if she becomes infected right before the labor, usually, the mother has to be isolated from the baby in order not to expose the baby. So there are a lot of precautions that are being taken, but as I mentioned, pregnancy is a risk factor for more severe COVID as well as pregnancy complications. So it's something to be avoided. And I wrote an op-ed about this, but it's really important to vaccinate people who are pregnant.
Akiko, but if I may add to these questions, I think media is providing sort of controversial points of view on this. I don't know how, but United States, I think in other countries, there are claims that actually pregnancy is not a risk factor. So how strong is evidence? I mean, is it something new? Was it also controversial in the States at some point or not?
Well, there were other controversies that took over. For example, the mRNA vaccines causing infertility and all kinds of misinformation. But the pregnancy being associated with higher risk for more severe COVID is well established, I think, by now. So that's no longer controversial. It's the sort of fear of vaccine during pregnancy that is still very deeply rooted in this country. I don't know about other countries in Russia, like what people are feeling about the vaccines and hesitancy, but it's certainly here very strong in the pregnant population.
Okay, thank you. Other questions? If not, Akiko, let me thank you very, very much again. And from all of us.
Thank you very much.
So please come in person as we discussed. You should stop by when you will be flying to Japan. Stop.
Okay. I'll use a parachute or something. Stop by. Thank you.
Okay. Thank you.
Thank you. Bye. Take care.